Hello, bio students. So tonight I'm going to be talking to you guys about something called the phospholipid bilayer. Wait, phospholipid. There we go. Hey, Abigail, do you remember the phospholipid bilayer? You didn't? Wow, that's really weird. The phospholipid bilayer is the fundamental reason why cells can exist. And I want you to think about this for a moment. Imagine you have a crazy outward environment. There's all sorts of things going on. There's daggers. Okay, they aren't really daggers, but imagine there are chemical dagger-like things. There's chemical explosions going on out here. And there's all sorts of nasty things out here in our outward environment that will break you apart if you are a molecule. So wouldn't it be great if you could protect yourself, sort of put yourself inside a suit of armor of sorts? And that's exactly what a phospholipid bilayer allows you to do. It essentially takes a set of molecules and it creates a fat bubble. So if you're hanging out inside of there, you're safe and happy, and there can be all sorts of chaos going on trying to attack you from out here, but you're able to maintain a nice cozy place inside here because you're safe in your little fat bubble. And that's exactly what the phospholipid bilayer is. It's a bubble of fat. So let's shift over here for a moment. Let's go over here. Don't forget that a phospholipid is a fat. Okay, so phospholipid equals a fat. And more specifically, it's a fat that has a couple of fatty acids associated with it, and a glycerol and phosphate head. And it looks like this. Let me go ahead and stop this for a moment and add in a picture. All right, there we go. This is a phospholipid, and you can notice here that the reason why it's a phospholipid because it has a phosphate group with it. However, it also has this glycerol backbone and two fatty acids hanging out right there. Now, these fatty acid chains, it's kind of funny because they're showing them both as being straight chain, sort of like stearic acid, but in fact, that is not entirely accurate. I probably should have found a better picture here because one of these actually is not fully saturated, and as a result, it has a kink in it right there, and it causes one of these fatty acids to bend, the other to be straight. And that's going to be a very important piece because, as you well know, if you have those kinks, then these phospholipids are going to be liquid at room temperature. And we want them to be somewhat liquid-like, as you'll see moving forward. So let me jump right over to here. Oh, shoot. Thanks, bubbies. And I'm going to heavily schematicize a phospholipid by showing here, this is my phosphate head. Okay, I'm just going to put a P there. It's really PO43 minus, but we'll make it easy. And then we have a lipid tail, or a fatty acid tail, and one with a little kink in it. So imagine we were to replicate a whole bunch of these things. Let me go ahead and do that right now. Okay, so what's really important here with this sort of group of soldiers, these phospholipids, is that one side of this phospholipid is really water loving. And that's this part, the phosphate head. It loves water. Okay? So we have water molecules cruising around up here in contact with the phosphate head. However, the lipid tails, just like all fats, hate water. And so they want to face away from the water. Now, of course, one of the problems you have here is that water surrounds the entire environment of a cell. So the only way that you can avoid the water is if you have lipid tails facing in next to lipid tails. So let me pause this for a moment and show you what I'm going to do. Look, isn't that unfair that I can copy and paste like that? It seems uncool, but that's what I get to do, suckers. So if you can imagine this, I have down here water on this side too. But there's no water here because these things, this area that I'm marking out in here is hydrophobic. Water hating. Hydrophobic. Whereas the other end, the phosphate heads on both sides, these are hydrophilic. Water loving. I hope I spelled that right. I wonder if there are two L's in philic. Oh well, I'll have to deal with that. So here we have a situation that's cool and all, but of course, isn't there water right here? Well, absolutely, you could imagine there'd be water right there. But imagine doing this. Let's scoot over to right here. Okay. Imagine this situation. Here I have a whole bunch of phosphate heads. Okay. Yeah. Oh, sweetie, I really can't. Does mommy absolutely need to talk to me? Okay. I'll tell her that I'm trying to do a screencast right now. Okay. And she still needs to talk to me? Oh, I see. I see. Okay. Imagine you had this long line of phospholipids. Well, you could imagine that perhaps this would actually connect to this. And instead of looking like what I have right there, I have a situation like this. A whole bunch of phosphate heads, and facing inward would be all their fatty acid tails. Now, of course, these are all double tails, so I'm being a little bit naughty in that I'm not doing double tails, I'm only doing single. But now you have a situation where all the hydrophilic pieces are in this area that I'm coloring in green right here, and all the hydrophobic are in there, and all the hydrophilic pieces are facing out here, loving the water, right? Cool, right? Now imagine this. This thing gets so big, it's not just this short, it's much longer than that, so that you have a situation that looks something like this. I'm going to go ahead and draw a really long one of these dudes. Okay, and it's going to go here, and then like this. Okay, and it's a little tricky here, but notice that I've basically created that round one that I have over there on the right, but I've just extended it into sort of this long worm-like structure. And, oh, Schaefer's texting me. Okay, like so. So again, it's just like this one, but I've elongated it, right? So I have, if you look over here, zoom in right here, I have phosphate heads coming around the corner, and the tails are pointing in like this, all sort of cramped in there. And then over here, same thing. Heads and tails all sticking in there like that. So it's just sort of like an elongated version of this dude right there. Now imagine that this connected to that end. And now what you're having is the precursor to what ultimately will be a very rudimentary cell-like structure which would isolate, just like we showed here, an interior space from an exterior space of water. So let me pause here and show you what I'm drawing. I'll draw it quickly and then you'll have to see what I've done. Take a look at that! How cool is this? So again, all I did was I brought these two ends and connected them together to make something like this. Now I have an internal watery environment that is separate from the external watery environment out here. So that means I can have a whole bunch of chemical reactions that are happening inside here that are not happening outside here, and vice versa. So when you can isolate a pocket of water and have certain chemical reactions happening on one side that don't happen on the other, then you start to have something pretty interesting. So let me move this now to the next slide and pause for a moment. My kids are fighting, so I have to tell them to shush. Hold on. All right, what we have here is a much nicer drawing that I did not do, showing the phospholipid membrane. And you should note here that these little balls here, that's a phosphate head. What happened? How come I can't draw on it? There we go, phosphate head. And these little tails that we got going here that I'll just highlight in yellow. There's one, there's another. And they have a little kink in it, so it makes them somewhat liquid. Oh, shoot. Penny Post is calling me to go up to the dining hall. I'll finish this in a second. I really have no idea where I left off because Penny Post came home and we went to dinner. So this is showing you a plasma membrane. But understand that the plasma membrane is what surrounds an entire cell. And in fact, yes, Lumpy? Can you pause it? Hold on. Yeah. All right. So yeah. No, I can't. And let's see. Oh, here, this is a good place to insert the next thing. I want to show you an actual micrograph here of the plasma membrane. So this is kind of cool. because you can see where it's pointing it, and right there, that's the plasma membrane. You can see it's a phospholipid bilayer, just like the phospholipid bilayer that you see right here. Right, there's two of these things, so I can go ahead and put in another right there. But in the plasma, oh, and you should also keep in mind that every single organelle that you see inside of a, no, that's a, yeah, every single organelle that you see inside of a cell is going to be made up of this phospholipid bilayer. So you have the bilayer that surrounds the entire cell and bounds the entire cell, and then you have also bilayers inside that make up things like the mitochondrion, the lysosome, central vacuole, endoplasmic reticulum, all sorts of fun things. But I want to draw your attention to this because this cell membrane or plasma membrane it has a lot of stuff going on here. It's not just the phospholipids. And as a result of all these other things in there, we actually have a way of describing this model of the plasma membrane. It's called the fluid mosaic model. Fluid mosaic, and it's a mosaic just like the mosaic tile murals that were made in Italy. Fluid mosaic model. And if you think about the tiles all being different colors, well, it's not that there's so much different colors that we have going on here, it's there are different shapes of different tiles. Let me point out a couple of key little pieces here. I'm going to use, do I have a pointer? I do, sweet. Okay, so this thing right here, is it actually pointing? There you go, yeah. Those little orange beans here, those are cholesterols. Remember how I talked about cholesterols are really important? Well, what they do is they allow this whole phospholipid membrane to be slightly more fluid in nature. Because in fact, it is a fluid. It's more fluid than it is a solid, which is what describes the concept of fluid mosaic model. And what that means is if you were to sort of zoom in on any of these spots, let's say that we looked at this, the head of the phosphate head of this phospholipid. Well, if I waited 5 minutes, it's very likely that this head will actually travel to over here. So nothing stays fixed. Actually, that's not entirely true. There's some things that are somewhat more fixed than others. But keep in mind that for the most part, all these things are moving around kind of like buoys in a large sea. All right, then you can also see that there are some interesting bits and pieces here. This is actually a channel membrane, and you can see this is a protein that actually will have an active site here for some sort of substrate that's flowing through there. And oops, I should erase that little bit there. And then you can see here, remember I talked about this some time ago when we talked about the big four biomolecules. Those glycoproteins, oligosaccharides of the glycoprotein, are sort of like ID badges for this particular cell. So anyway, you can see there's a whole bunch of things in here. They look like potatoes floating in there. They're, for the most part, they're proteins. But fundamentally, the basic material are these phospholipids, and they are cool. All right, let's see if there's anything else I need to pass. No, I don't think so. I'm going to freeze the frame on this. Bye, all.